What I would like to do today is to tell you a little bit about the work we're doing to understand how environmental exposures, especially heat stress and also drinking water contaminants may affect our kidney health. And trying to give you a glimpse of the work we're trying to do from an epidemiological perspective and also from a mechanistic toxicology perspective. And really have us all leave the seminar hopefully thinking about or more deeply about the role of the environment in kidney health, because I think that remains one of the really underappreciated areas when we think about our kidneys. So as you can tell from the title, we are working primarily in farming communities, and as I say, we only have emerging lessons for now. So hopefully there will be more questions that would inspire some of the work that we could do in the future. So to get started off, I will first say that we think a lot about how the environment is changing around us, especially with increasing pollution, and also especially with climate change that's leading to particularly these increasing intensities of heat stress and heat waves that is affecting communities around the world. So are they affecting, or do they have an effect on our kidneys? And that's sort of the overall theme that I want to focus on. Before I do that, I want to highlight that kidney diseases are a significant burden in the United States and also globally. So right now, there are about 800 million people who are at risk of kidney diseases in the world. Either they're diagnosed with chronic diseases or chronic kidney diseases, or are on their way to getting diagnosed, where they have some markers of poor kidney health already. And in the United States, about 1 in 7 people, that's about 14% of the population, in other words, 35 million people, are estimated to have chronic kidney diseases already. And about 1.2 million people die from kidney failure in the US. And that was data from 2023, and it's increasing, unfortunately. Currently, kidney diseases are placed as the 12th leading cause of death globally. And the projections are that it will become the 5th leading cause of death by 2040. Except that these projections actually don't, they only take into account some of the typical risk factors that are that can affect kidney health, such as hypertension or diabetes or aging. And as I mentioned, there's been an underappreciation of how the environment might be playing a role in either directly affecting kidneys or exacerbating or accelerating poor kidney health conditions from the comorbidities that are already affecting these communities. So why are kidneys so vulnerable to or sensitive to environmental change? And I think about the kidney kind of as the garbage processing center in the body, where they filter through about 200 liters of blood every day. And from that, about 2 liters of water is excreted out, which include the biological waste that we produce, like creatinine, resulting from muscle breakdown, or toxins that we're exposed to daily, or the medications that we take. So, for those of you who are not looking at pictures of the kidney every day, this is what they look like. And what you can see in this figure is in red. It's the blood that's coming into the kidney with toxins and waste, and they get filtered through the glomeruli. And then some of that filtrate will be reabsorbed through the tubules, especially really important biological macromolecules, including some low molecular weight proteins. And if everything works well, you're holding on to all your proteins and you're only releasing some of that waste product that will be added into the urine and that's excreted out. And as you can imagine, on a given day, kidneys are filtering pretty much everything that we are inhaling or we are ingesting, unless they're excreted through our feces or through respiration. So kidneys are generally exposed to a large number of environmental contaminants, even though they may be at lower levels, right? So in parallel, we are also seeing that kidneys might be sensitive to heat exposure. And the sort of catchy term that people have used is that it's a suffocating organ because the inner kidney operates under very low oxygen conditions. I won't go into too much depth in trying to explain this, but basically the way that kidney vascularization works is that there's a lot more oxygen that is required to maintain some of the highly energy intensive protein activities like sodium, potassium, ATPase. And because of that, a lot of oxygen are retained or there's a high oxygen tension in the outer cortex of the kidney, whereas as you get into the deeper and deeper kidney, there's a lot smaller or a lot lower oxygen tension. And this is fine under normal conditions and works well and helps us concentrate the urine and really get rid of the toxins that we might be exposed to. Where this begin to matter is when we are exposed to a heat exposure. This is because, as you may remember from biology a long time ago, hopefully, is when we heat up, we vasodilate. So we bring blood vessels close to our skin and improve circulation to our skin and to other extremities to dissipate heat. And this reduces even the little oxygen that's coming into the deeper kidney, which can lead to this increasing susceptibility to ischemic injury or low oxygen-induced injury. So this coupled with loss of water and electrolytes from sweating, which adds a further burden, we are beginning to see that during heat wave events, for example, there's a 10 to 25% higher incidence of acute kidney injury-related hospitalizations in the United States again. We don't have such data actually for other parts of the world as clearly. And there's generally been considered that there's about a 1.6-fold higher odds for an episode of acute kidney injury for a person when the temperature in the region that they live in goes outside of their thermal normative or the normal temperatures that they typically experience. So these epidata are indicating to us that acute kidney injuries and kidney health might be compromised under heat wave or heat exposure conditions and scenarios. So just to summarize that bit a little bit, basically a low oxygen insult, a direct heat injury, and dehydration coupled with exposure to even very low levels of chemical toxins, which on their own might be totally fine. All of that may exacerbate risk from other comorbidities for our kidneys. These different environmental variables may have synergistic interactions. And also there's data showing that some of these can directly lead to acute kidney injury, all of which can contribute towards chronic kidney disease development. And we wrote a short commentary on this with Dr. Sanders at University of Pittsburgh. So I just wanted to highlight that because we are beginning to see that AKI is emerging or acute kidney injury is emerging as a concern in the United States. Right now, about 13 million people suffer from AKI around the world, and with about 10% in the US. And the overall data indicate to us that perhaps only about 50% of the people can actually recover from that AKI and undergo repair, where the others might actually end up progressing into CKD status. So AKI or repeated AKI can actually lead to potential chronic kidney disease outcomes. And all of that has somewhat culminated in this emergence of a disease called chronic kidney disease of uncertain origins or non-traditional origins, CKDU or CKD-NT. There are several other names for this if you're already familiar with it. Around the world where communities are diagnosed, people living in communities are diagnosed with chronic kidney failure, but they don't have any of the other typical risk factors that you see that are associated with kidney diseases, other than exposures to environmental contaminants and also potentially to heat exposure. So I assume some of you are familiar with CKDU. It's been called a quintessential climate disease and also a sentinel disease of environmental change in the New England Journal of Medicine. Also received a lot of attention, media attention, including our work in Sri Lanka, all suggesting that this disease is affecting communities that didn't used to experience this type of chronic disease outcomes in the past and is having devastating impacts on those communities. So what I want to do today is to tell you a bit about the epidemiology of the disease and the work that we are doing to understand both the markers of the disease and also try to get at the mechanisms of where this disease might be originating or initiating in the kidney, both from the model animal work and also from some population health work. Just to give you some numbers, CKDU typically are diagnosed in younger people than the typical CKD that you see, for example, in the US or in the global north. In these cases, in CKDU cases, you see people as young as 35 on dialysis and then eventually don't make it more than another 3-4 years in life. And as I said, they're not associated with any other usual risk factors, especially diabetes or hypertension. And often men are impacted, and this might be primarily an occupational preference that might be playing a role here, but there could be other biological reasons as well. And this is known as the widowmaker disease, for example, because men are at young ages being impacted by this. Just to give you some numbers, 1 in 3 men are at end-stage renal failure in this one community that's impacted in Nicaragua. There's one community I visited and did a little bit of work in Udanam in India, where 60% of the men are diagnosed with CKDU. There's been a 5-fold increase in a number of communities over the last 20 years in some of the impacted countries in Mesoamerica. And Sri Lanka, where we do most of our work, at a given time, there's about 20,000 people who are seeking dialysis treatment because they're at end-stage renal failure. So it's affecting communities in a really profound way. And we are beginning to see that hotspots are emerging in the tropics around the world, especially in agricultural communities. So there are really well-established hotspots in Sri Lanka, in India, in a number of Mesoamerican countries, especially Nicaragua, Guatemala, and El Salvador. And there are hotspots that are beginning to emerge in several countries in Africa. We are starting a cohort in Kenya right now. Potentially also an emergence in Southeast Asia. And then also there is suspected cases in Texas, in North Carolina, and there are some cohorts that are being followed also in California, particularly among migrant farm workers, to see if this prevalence of CKDU is emerging and begin to emerge further and further north of the equator as well. So what's causing this, or what are the risk factors that people have identified over the last 20 to 30 years of doing this work? The 2 major hypotheses are that it's associated with exposure to heat stress under strenuous labor conditions that many of these farmers are engaged in, or it's associated with environmental contaminant exposures, such as drinking water contamination that can happen from having your drinking water well right next to your paddy field that uses a lot of agrochemicals, or direct exposure to agrochemicals. Fluoride in the drinking water has also been shown to be associated with this hard water, and potentially some microbial products in the drinking water as well. And then also potential exposures to metals in the water, and also potentially in the household dusts. But a particular element that's gained a lot of attention is silica, where silica nanoparticles can be created from burning some of these agricultural wastes, like straws from rice fields. And there's been studies showing that silica may also be playing a role in this. So there's quite a bit of hypotheses and studies that have come up. What I want to show you is the work that we've done to support some of these hypotheses. It doesn't necessarily mean that the others are not correct. What I do want to say is that what all of this work, and if you leave today without listening to the rest of the talk, what all of this work is really highlighting around the world from all the researchers engaged in these studies is that there are a number of environmental contaminants and exposures, such as heat stress, that do present a risk to our kidney health. And we're really barely scratching the surface of our understanding of that. So to share our work, I want to take you to Sri Lanka, where our primary field stations are set up. So I'll talk a little bit about why Sri Lanka, and then talk a bit about some work we've done with communities there both to collect biological samples and also environmental samples, and our in vitro work, and then what we are doing now in some of the communities by going back to those communities based on the data we've collected in the lab. So this is Sri Lanka. It's a small island in the Indian Ocean. It's a very beautiful place. I'm not biased, but I was born there and I grew up in this tiny little town called Gaul, where there is no CKDU. And what you're seeing in this map is the prevalence of this disease. This is not the most accurate map. There's a number of issues with it, but it gives you a good picture around where the disease incidences are found and where it's not found. And in blue, I'm highlighting regions of the country where CKDU is not detected. And in these dark red areas, you're seeing areas where there are some significant hotspots. And the two green dots here are two areas we've done a lot of work around in. And other than being able to understand the language and have an affiliation with the country, Sri Lanka does present a good case study for this work. They have a relatively highly organized public health system. I say relatively because compared to many low middle income countries, there's a lot of data that you could actually gather over the last 30 to 40 years. And there's a lot of public awareness and high literacy rates, so you can work in the fields where the communities are on board with these kinds of studies. And the government has prioritized CKDU as one of their major public health crises. So you can get a lot of help from the government sources. And there's, because of that, many resources are allocated for this. And Sri Lanka, in a very small space, it's essentially about 200 to 300 miles wide in the Logness Point. So it's a pretty small geographical space you're thinking about, and provide a number of climatic zones and agricultural zones where you can do these comparative studies to better understand what are some of the environmental variables associated with disease emergence. So just to quickly show you a few pictures from our field site, this is what it looks like. These two are two incredible community leaders that work with us. In green t-shirt, Chamal Priyanta is a physician. He's from the village. His mom died from CKDU, so he's really invested in this work. And Mr. Saman is another a great collaborator with us who's been working with us for the last 5 to 6 years. So I just wanted to say a quick note about we couldn't have done any of the work that we're doing without the support from these folks. So what are we trying to do with these communities? So one thing we've done is to collect urine samples from our CKDU patients and control farmers who live in another part of the country and try and understand what does the urinary proteome look like. Fairly straightforward study, but this actually gave us a lot of insights into what to think about next. And this was work done by Dr. Ramya Kohli, who was a postdoc in the lab. And we had just a very few cases, about 25 cases and controls. But like I said, we were able to identify pretty striking differences between our populations. We found that there's a number of proteins that have increased in abundance in CKDU patients, about 230 proteins in the urine, and then about 700 proteins are decreased in abundance. In other words, the actual total protein levels have gone down in the patients compared to the control groups. When we look at the origin of these proteins, we find that in the order of abundance, we find that many of the proteins that are ending up in the urine are originated in the proximal tubule and then the distal tubule, and also from the collecting duct, which is suggesting to us that there is some kind of damage that's going on in the tubular regions of the kidney as opposed to some of the other regions. So what's changing, and one thing I want to highlight is that when we look at the number of proteins that are increased in abundance in our patients, majority of those proteins are low molecular weight proteins, and low molecular weight proteinuria is a sign of impaired protein reabsorption, which suggests to us that there is a proximal tubular dysfunction that's going on. So this is helping us to think about which part of the kidney might be affected based on this proteomic work. And when we look at a little bit deeper, what are some of those proteins that are increased in abundance? What we find is that the proteins that are higher in our patients are proteins that can bind to other proteins that are involved in protein reabsorption that have decreased in abundance. So just to explain that a bit further, what you're seeing here is in our control individuals, these megalin and cuburin proteins that are involved in protein reabsorption are present at the same level as some of these ligands that can bind to those megalins and cuburins, like retinol binding protein or transferrin. But when we look in our patients, we see this very clear signature of decrease in megalin and cubumin and an increase in these retinol binding proteins, which further really confirms us that there is a defective protein reabsorption or impaired protein reabsorption that's going on in the proximal tubule in these patients. The other sort of major finding for us was that we found that proteins associated with lysosomes was one of the most significantly enriched, or it was the highest enriched pathway in the entire proteome. What that is corresponding to is this other data sets that have come out looking at kidney biopsies from CKDU patients from Sri Lanka, which suggests that there is a potential lysosomal disorder that might be underlying this disease, where they are able to detect these single membrane-bound inclusion bodies in the proximal tubules of the CKD impacted patients, which would explain the proteomic data that we are seeing, which sort of underlies this hypothesis that several folks have presented that lysosomal dysfunction might be a hallmark of CKDU compared to CKD and some of the other different types of CKDs. The second major pathway that was enriched was metabolic pathways, and especially proteins involved in mitochondrial function. And when we look across our data set and also other omics data sets coming from other CKDU impacted regions in the world, we find that mitochondrial function or changes in cellular metabolism is a key signature of CKDU patients compared to people who don't have the disease or who are potentially on their way to get the disease but don't have the full kidney failure diagnosed yet. Now, obviously, the big caveat is the mitochondrial dysfunction a cause or an effect, right? And one thing to support that it's potentially a causative change is when we look at our mitochondrial proteomic signature, or our overall proteomic signature rather, that proteomic signature in our patients in the urine is most similar to the proteomic signature of patients with mitochondrial diseases. Some of them have kidney diseases, some of them don't, but generally, this really nice paper in 2015 showed a list of proteins that are abundant or differentially abundant in urine of patients with mitochondrial diseases. And when we compare our proteome with that proteome compared to some of the other proteomes of kidney disease patients, we find that our CKDU patient proteome is most similarly related to patients with mitochondrial diseases, which is suggesting to us that perhaps there is some underlying etiological cause for mitochondria and also potentially for lysosomal dysfunction. So I'll talk about that in a little bit. What we are doing now is, and this is why I'm also hoping that there may be folks in the audience who would want to talk about some of the work that could be done in this context, and I'd love some advice as well, is Sri Lanka just built what they call the largest nephrology care unit in all of South Asia, because they have to meet this really high demand for dialysis for patients coming from all of these hotspots. And we now have a collaboration set up with them where we are collecting kidney biopsies and blood and urine from patients who are presenting at different stages of CKDU and also CKD. And I'd love to hear any thoughts on what some of the suggestions you may have on how to what to look for in these biospecimens that we are collecting. So I want to now move on to showing a couple of slides on what we are doing to try and understand what are chemical exposure risks that might be associated with CKDU incidence in terms of environmental contaminants. And we did several studies. One study is to put wristbands in our farmers and our controls, and also people who live in non-CKDU areas, to try and understand what are some of the volatilized organic compounds in the in the regions are that we could then analyze to identify potential chemical contaminants they're exposed to. And this is a collaboration with Professor Heather Stapleton at Duke and also another student, Sakina Shahid. I won't show that data, but I just want to highlight that we are trying to understand aerosolized contaminants through this approach. The second part we focused heavily on is through a collaboration with Professor Lee Ferguson, who's also at Duke, and Jake Ulrich, to conduct a pretty thorough drinking water analysis for the impacted communities. And this is Samir Gunasekara, who's a postdoc working in this project, collecting a water sample from a dug well. This is one of the nicest wells I've seen, but it's right next to this rice field that's being irrigated, but also getting fertilized and pesticides added. And in a nutshell, we do measure those signatures in the drinking water samples. So I'll show you one study we've done in this space where we think that water can both serve as a source of exposure for the community members, but also it shows us a snapshot of what's in the environment in these communities at a given time. So these are some of the sources. We've done hundreds of water samples. And just to put it all into one slide, Jake Ulrich published this work a couple of years ago, where we found that in patients who are diagnosed with CKDU, in their drinking water, there are metals and metalloids and fluoride and glyphosate that are found in their drinking water samples, and especially glyphosate, fluoride, water hardness, and vanadium are some of the most highly positively associated with chemical contaminants, highly positively associated with CKDU prevalence. I think we all know what glyphosate is. There's been a study that just came out a few weeks ago showing that there is NHANES-based data analysis that showed there's an association with glyphosate exposure and decline in kidney function. It's the active ingredient in Roundup, and it's one of the most heavily used herbicides in the US and around the world. So we wanted to follow up a little bit on this, especially because what we found in terms of chemical signatures, nothing was at high enough levels that could cause an alarm. And in fact, everything was way below the EPA permissible limits. So the question to us was, is this a mixture effect that we should be thinking about? So this is when our in vitro studies came into the picture, where we took these water samples and did a series of exposure studies to understand both kidney developmental effects of exposure to these chemicals that we find in the environment, and also long-term chronic kidney health outcomes of exposure. And for this work, we used the zebrafish model, which has emerged as a really high throughput whole animal system to try and understand organ health outcomes as well as other toxicological outcomes. And in the space of the kidney, zebrafish, they do have a kidney. I sometimes get that question. And they share pronephros that are similar in cell types with human nephrons. Their glomerular filtration barrier is ultrastructurally pretty identical. There are slight changes, but generally it's a very good kidney development model to understand kidney injury, especially during fish development. We can get data to inform human health questions, but also we could do adult exposure studies to better understand how chronic exposures may affect kidney function. And the big advantage of zebrafish compared to some of the other models is that they breed in large numbers. You can keep them in the lab in a very small space and do many different types of exposure assays in short and long periods of time without really taking up a lot of resources. And also because they're transparent early on, you could actually visualize some of the changes that might be happening in the kidney. So one thing we did was to try and understand which chemicals should we expose our fish to based on our environmental sampling data. And when we look at the total molecular burden presented from the chemicals in a drinking water sample, we found, and this is in parallel to the data I showed earlier, we found that glyphosate is one of the most prominent elements in the drinking water samples, but also vanadium, arsenic, cadmium, and lead. Again, they're all at very low levels. You're safe to drink all of this, including the glyphosate under the current EPA guidelines. And we find them in the drinking water samples. And the molecular burden presented by these elements are higher in the CKDU endemic regions compared to the non-endemic regions. So we created a mixture that contains this metal glyphosate complex and also glyphosate alone and the metals alone. And we did developmental exposure studies and also chronic exposure studies with our zebrafish model. And this is our exposure paradigm for the adult study where we did 0, 10, actually 30 and also 60-day exposures. And this is work by Ilaria Merutka, who's a PhD student in the lab, and also a former PhD student who just graduated, Remy Babbage. So I'll show you some zebrafish data in the next 10 minutes, and then have 5 minutes or so to talk about some of the other data sets we have. Just to quickly highlight some of the tools we are using. One assay that we could do is to look at kidney function. And this is to actually measure low molecular weight proteinuria using this transgenic line that we received from Dr. Lennon, where we can basically measure the level of this nanoluciferase in a plate. And the more nanoluciferase you're detecting, that's indicative of proximal tubular damage and low molecular weight proteinuria. So when we look at the total luminescence in our plate following extraction of water from the tank that the fish are living in, where they were initially exposed to these chemical contaminants, what you see is that early on, there's not a big difference. There's some animals with our metal exposure or glyphosate exposure or metals and glyphosate exposure. Some animals begin to show some increase in proteinuria. This somewhat subsides within about 20 days. But when we look at 60-day time courses, we begin to see several animals showing this low molecular weight proteinuria. So this data are not significant. So we're adding a lot more data points to this, but this is a very promising result to show that both the metals alone and the glyphosate alone, but especially the mixture, can really lead to injury to or potential changes to the proximal tubular region of the kidney in the zebrafish. And we wanted to dig into this further to better understand, is there an energetic reason for this, especially given the data we were finding in the patient proteome with mitochondrial proteins in there. So we developed a method to measure whole kidney mitochondrial function. So we can take out a kidney from a fish and then run it using this seahorse bioanalyzer assay that we have in the lab, where we can do ex vivo tissue respirometry. And what we find is that mitochondrial function is reduced, especially at 60 days, but also at 10 days in animals that were exposed to these chemicals. Again, few data points. And like I said, we are ramping up this study more because this is working, and it's also promising that well, it's not promising for the fish or the people, but it's promising that we are seeing mitochondrial dysfunction going on. We looked at mitochondrial DNA copy number in these kidneys to see, is it that there are fewer copies of mitochondria with exposure? And we don't find that to be the case, which is telling us that the mitochondria are there, but they're just working more inefficiently with these exposures, especially with the glyphosate and metal exposure. One other endpoint we looked at was mitophagy. So this is when mitochondria try to get rid of damaged mitochondria on its own. And we can measure the number of mitophagic area in a given proximal tubule using this MitoQC transgenic line that we got as a gift from Dr. Tristan North. And again, we see that there's an increase in mitophagy with metal and glyphosate exposure, but also with metal exposure alone and to some degree with glyphosate exposure alone. Excuse me. So this is 10 days. And when we look at this over 30 days, we see, again, some subsiding of these effects. But what we really see where these trends start to pop up is with 10 days, you begin to see an increase in mitophagy. And with 60 days, you're beginning to see a decrease in mitophagy, which is telling us that mitophagic events are taking place early on, essentially as an adaptive response to the chemical insult. But once the exposure is more chronic, then you begin to see a decrease in mitophagy, suggesting that there's potentially pathological condition that's kicking in the kidney at this point, especially in the proximal tubules. This change in mitochondrial function was further confirmed with some of the metabolomics work we've done, which again showed to us that there's a number of processes involved in mitochondrial metabolism and also fatty acid metabolism are some of the most enriched pathways that are differentially abundant in fish exposed to glyphosate and metals, especially a little less so in the metals alone or the glyphosate alone group. Um, Looking at some transcriptomic data again with these exposures, and this is really early on in development, so very short exposure to get a better sense of what are the potential molecular initiating events that might be undergoing with these exposures. We see that our principal component analysis clearly separate out our controls, thankfully, but some overlap between glyphosate and then also glyphosate and metal groups, but metals stand out completely differently. And this is something we see with the entire data set, where glyphosate alone is not super toxic to the kidney or the genes that are changing. That's not the right word to use, but it's not that it doesn't seem to be bad on its own. But when we mix it with the metals is when we begin to see some really clear phenotypes showing up where kidney injury might be starting with those exposures. And one of the other important changes in gene expression patterns we saw with that data was also change in lysosomal function and also lysosomal integrity with these exposures, which again relates back to what we were finding with the patient proteome and then other groups are finding in the kidney biopsies from the patients. So what this is telling us is that glyphosate in combination with metals may lead to lysosomal dysfunction and also mitochondrial dysfunction in the zebrafish kidney. And we confirmed this using some histopathology analysis as well. So I'll skip that real quick and say that we are following up on this work in collaboration with the group that's here led by Dr. Alison Sanders through the NIHS-funded Victor grant, and some really neat tools are being developed in this to compare our zebrafish data with mouse data with 3D organoid data that are done in Neil Hackreday's lab at University of Pittsburgh, and Jackie Ho there is doing the mouse work. So really excited for that collaboration to move forward because we are finding some really important parallels between our model systems, but also learning about limitations and advantages of each of the models. And I just want to quickly also thank Ian Drummond, who really got me thinking about the zebrafish model for kidney studies. I want to quickly touch on a couple of other studies we're doing in the next few minutes. One is that because we're finding that our zebrafish model is showing kidney development is affected from these exposures, children living in these communities drink the same water. They drink the same water as their parents do. So we wanted to screen for kidney health across pediatric populations in CKDU impacted regions versus unimpacted regions. And so just to show you some of that pediatric data, I just have one slide to show that we were finding that there is an increase in urinary kidney injury marker, KIM1 marker, in villages that are endemic to CKDU, and also villages where we are seeing an emergence of CKDU. But that trend didn't really hold up for the non-CKDU villages. In the CKDU villages, we already find about 6 to 10% of the kids already show a higher abundance of KIM1 or kidney injury molecule 1, which is indicative of proximal tubular injury in the kids in these regions. So we did a meta-analysis of all the data around the world that have looked at pediatric kidney function in what we call high exposure or CKDU-like exposure communities, and all of that globally suggests to us that kidney injury is prevalent in kids living in some of these agricultural communities around the world. And this is currently under review and led by a nephrologist here at Duke, Dr. Anna Strasma. So we're following up on this with a 3,500 children cohort in Sri Lanka right now that represent multiple agricultural regions and also climate regions. That would help us understand, is this kidney injury something that just disappears over time? So we hope to follow up these kids for the next 5 to 10 years, depending on how funding works out, but also we have some funding for now to keep this cohort going. So I'm grateful for that. And also to better understand which of the environmental variables better explain the kidney injury that might be going on in these kids. And one other last study that I want to share really quickly is to think about heat stress. So everything I've showed so far suggested to us that environmental contaminants do present a risk to kidney, and we have some mechanistic understanding of what might be going on, which is really giving us a lot of new areas to pursue to think about how each of these contaminants, both in mixture and also individually, may affect kidney development and kidney function, and use some of that as markers to better understand pediatric kidney health in the affected regions. What's puzzling, though, is that we don't necessarily find CKDU is explained only by exposure to environmental contaminants. And this has been shown also in South America and also in Sri Lanka. And the other prevailing hypothesis is that it's not just agrochemicals, it's also exposure to heat stress. So we wanted to explore this a bit further, especially with these two postdocs in the lab, where we looked at just snapshot kidney function across multiple communities around the country that live in different climatic zones. So if you're in this really yellow area and you're a farmer there, you're working really hard, but in a really nice, cool, sort of almost like the British fall weather type weather conditions. And then you have the salt pan workers who break salt for a living to sell in the market. They are engaged in some of the most strenuous labor activities I've seen in some of the hottest parts of the country with no shade. And then we also looked at kidney function in fishermen who live in also some of the hottest parts of the country, where these fishermen don't have motorboats. They actually row out into sea, and they don't have access to fresh water during the day other than the little bottle they carry. So they're also working very hard, but out at sea. And then also our farmers who live in the CKDU impacted region. So we compared kidney function across all of these different groups. And what we found was that the probability of an individual with already declined kidney health was highest in the salt pan workers who work in the hottest parts of the country. So what you're seeing in this figure is this is EGFR, kidney filtration rate. And then along the x-axis, you're seeing each of the occupational groups from different parts of the country, where as you go more towards the right or to my right, you're seeing people who live in cooler and cooler regions. And what you're seeing is that there's this distribution of EGFR, and the lowest EGFRs are present among our salt pan workers, right next to our farmers who work in CKDU impacted regions. In the CKDU impacted regions right here, there are more individuals with really poor kidney health, but as a population, only about 6 to 8% of those individuals do. So in a nutshell, what we are seeing is that strenuous labor under high heat exposures can reduce your kidney filtration rates and can potentially lead to kidney dysfunction, but we don't find CKDU in our salt pan workers. We only find CKDU in the agricultural regions. One caveat I'd present to all this, though, is that we don't really have a good way to define heat stress for our farmers or for our salt pan workers. So before I conclude that heat exposure is associated with decline in kidney function, I want to show that there is a caveat in how we can actually measure and define heat stress. And I think there are many groups around the country are working on this kind of problems. And what I want to say is, especially in Sri Lanka, when you look at an average daily temperature derived from European climate data models, what you see in black is the thermal extremes. What you see in red are the on-the-ground measurements that we are making based on weather stations we've set up right at the location where the satellite data are derived. And we see a big discrepancy. Basically, it's saying to us that the European climate data that we use for our analyses actually are severely underestimating heat exposure compared to what we are measuring on the ground. And we want to now ground truth that we just got some funding to do this, so really excited to move this work forward. with wearable heat core temperature monitors that our farmers are wearing right now and engaged in field activities. So we can actually get data on their heart rate, core temperature, skin temperature, and correspond that with some of these satellite data that are coming through. So we can then correlate that with hopefully our kidney injury markers to see are we inaccurately defining heat stress, and also what is a physiologically relevant heat stress for a Sri Lankan farmer or a Sri Lankan soil pan worker in these conditions. And another sort of caveat to that is, what do we define as heat stress? Is it the change in average monthly maximum in temperature? Is it the variance, all the fluctuations that are going on? Is it the rate of increase in temperature? So when we look at Sri Lanka again, you see that there are some regions of the country that are warming a lot faster than some of the other regions. But when you look at average temperature, those regions are still hotter than some of these other regions. So is it the rate of change, or is it the average monthly change? Or is it the number of days that people are working under where they're exceeding the WHO thresholds for outdoor working conditions? So all of this are opening up a lot of questions. And I think I will again leave by saying we are barely scratching the surface of environmental drivers of kidney health. We have some hypothesis that there are potentially developmental and early childhood exposures to either heat or these contaminants that may increase susceptibility of the kidney to some of the adult exposures, or these continuum of exposures through life and interactions between these are actually what's leading to kidney failure in the CKDU communities, but also they might be contributing to that number of people who are diagnosed with CKD in the rest of the world. So we have some big questions and big gaps that we are thinking about. How do we define heat stress in the context of kidney health? How does heat exposure affect kidney physiological response to other toxins, including the medications that one might be taking? And how do we actually determine toxicity of multiple chemical contaminants, even if they're present at low levels? And this is my last slide, where I want to say that I think while our lab and as a researcher and an academic, I could really dive into some of these questions. I also think a lot about, do we know enough now to implement changes for the communities that are impacted, where we could actually hopefully mitigate some of the later life consequences that can happen from some of these childhood exposures that's ongoing. So with that, I want to thank all collaborators, students, and the wonderful community members who worked with us, and also the funding agencies that supported this work. And I'm happy to take any questions. Thank you again.
Really great presentation, Nishad. Thanks so much for walking through. We do have some time for questions, so folks can put it in the Q&A. Additionally, I'm not sure if people can ask if they can talk, but I can allow them to talk. So they can either ask for that or put it in the Q&A if they want to ask their question verbally instead of just typing it. You did a really good job, Nishad. I would write down a question and then 5 minutes later you would answer it. But I have some I can kick off with, and we can go from mine to the group to others. You talked a lot about the zebrafish work, and I think that's really exciting. And you talked at the beginning about sex differences. I know it's not the easiest to sex zebrafish in the beginning, but if you're doing adult exposures, is that a thought to try and maybe do, female exposures and male exposures?
Yes, really good question. I could spend another 20 minutes on this. So with the zebrafish model, that's actually what Ilaria has done, and that's why I only showed half the data to you, because initially we didn't expect large sex differences, but we kept finding that there are differences between male and female fish in our adult exposure studies. So that's another area to think a little bit deeply about for us now. So we are beefing up the data set with both sexes, with the hope of being able to either where the difference goes away or the difference becomes more prominent. So we can now start backtracking to figure out where that's coming from. We do see sex differences in our pediatric cohort as well. So generally in communities where children show about 6 to 10% of the children show an increase in KIM1, we see that primarily in male children and not female children. We also see that NGAL levels are higher in female children and not in male children. And increase in NGAL could potentially be interpreted as a protective mechanism against kidney injury, and baseline high levels may be doing that in our female children in these communities. The third part I'd add is that there's a group at Duke who's really clearly shown that in mice, I believe it's in mice, either cell culture or mice, that if the cells came from males or females, or whether it's in mice, that female specimens have a much better antioxidant defense response compared to male mice in the kidney. We also see very clear differences in EGFR in the mice from the data that Jackie Ho is developing at Pittsburgh. So I think there is a whole area to think about for this that I think, again, we're sort of scratching the surface around for now.
Yeah, really interesting. I see Peter's hands up. I'm going to allow Peter to talk. So hopefully you can now join us, Peter.
Can you hear me?
Yeah.
Yes.
Yeah, so really nice talk. Very fascinating stuff. So I'm just thinking in terms of like biomarkers for heat stress. So on the one hand, does this show up, say, if, in blood markers such as, a BUN to creatinine ratio,
Yeah.
which we typically use like in the ICU to evaluate dehydration, right?
Right.
So, on the other hand, are there like heat shock proteins that get excreted into the urine? Because it's not really a typical, urine biomarker for kidney damage, but in this case, if that's where, if the proximal tubules where you're suspecting a lot of damage is going on because of, you're talking about the low molecular weight proteinuria,
Right.
then does heat shock protein show up in the urine?
Short answer is no. We haven't found a clear signature of. There are some elevations, but when you look at enrichment and all the statistical cutoffs, I wouldn't feel comfortable saying heat shock protein does show up in our patients versus in our healthy individuals, which is a different analysis from, I think, what you're getting at, which is if you have a farmer working outside for, let's say, 7 days in the hot sun, and take urine samples on day one before they start the farming season, and then 3-4 days through the season. And then at the end, do you see a change in heat shock protein levels? And that's the grant we just got funded to do. So hopefully I'll have some answers on that. We have measured KIM-1 and also markers of inflammation. And this is tryptophan kinase ratios, and also malondialdehyde. Sorry,
MDA, yeah.
MDA, exactly. Levels as a marker of oxidative stress. So we can, so we are seeing that, these farmers, they have a break from farming for about 3 weeks, about 3 months, and then they start farming. So we have urine samples collected the morning of the farming season where they haven't done any farming. They just kind of live in the regions, exposure to general heat stress. And then we've collected 24 hours, 48 hours, or 3 days, and then 7 days, and then 1 month and 3 months. And we do see that increased inflammation and increased oxidative stress, or MDA levels, is as well as KIM-1 within the first few days. And then they all kind of recover back to normal levels over time. The caveat for now is that we don't know how to define heat stress for them. So they are working under hot sun. So we want to also figure out a better way to define this farmer was exposed to heat exposure and they had this level of elevation of KIM-1 through time. And another marker we will measure in the urine is heat shock proteins.
Thanks.
Yeah, thank you.
Thanks, Peter. We have one question in the chat, and that might take us to about time. So that's the question for me. It says anonymous attendee, which is a little ominous, but they asked, have you looked at lung capability or capacity? Is there a history of respiratory illness in kidney patients?
Yeah, this has come up a few times. I'd love to know who's asking the question. I don't have the, I should say, I don't know how to do it. Couple of respiratory physicians have been interested in looking at that exactly. We have the cohort, we could do it, but no, we haven't done that. And the two things that keep coming up is respiratory health and then also just basic neurobehavioral outcome measurements that you can make in these pediatric cohorts, because you are looking at a chronic, low level, but chronic organophosphate exposure in these communities. So those two things are up for grabs if anybody's interested.
Great. I don't see any others, and that was really a lot of talk and a lot of data to go through. I mean, I think, Nishad, if we can maybe share your contact info and people have follow-up or ideas for, specimens or idea for the kidney center, I think it would be great for them to get in touch with you.
Absolutely. Please share. I don't know if I put it on the beginning of my slides, but if you there's only one Nishad at Duke, I think. So you could find me easily.
Yeah, I'll happy to share it in the follow-up with the recording. So others might even reach out to you.
Yes. Thank you.
Well, great. Well, we're right at the top of the hour. I really appreciate the presentation and your time. This is really illuminating. So thank you so much. And thanks for those that tuned in. I hope you all have a great rest of your day.
Thank you so much.
All right. Take care. Thank you.